Hey everybody, this is my tutorial for grade 12 biology on the Sanger method of DNA sequencing, the concept of chain termination. And this is just one of the methods, probably the most famous method, at least the one that's taught in the grade 12 biology course in the molecular genetics strand here in Ontario, Canada, for how to sequence a strand of DNA. All right, so I've created a little sequence of flash images, flash animations, whatever, to do my best to illustrate this process to you, because I know it can be a little bit complicated. So imagine that we've got a virus here with some unknown DNA. So we've got a strand of DNA here, and we don't know what the order of the bases are. So we want to sequence this strand of DNA. So we got to get it out of the virus. So here it is. It's an unknown DNA sequence, and this represents the sugar phosphate backbone. And we, of course, don't know the order of bases. But in order to do this little activity here, we're going to have to pretend that we do, so we can show you how the thing works. So I'm going to reveal the actual bases here, and you can see that we've got 12 of them here. CTG, AGT, CAA, GTC. And this is a DNA strand, so these are all deoxyribose nucleotides. All right. Now you're going to notice that I've only got one strand. I don't have like a double helix of DNA here. And that's because we want to figure out the sequence of DNA on one strand. We don't care about the other strands. So we pretend that we separated the double helix apart. We're just trying to read one of them. Okay, so I'm going to hide that up again. So pretend that it is an unknown sequence. Now, in order to do the Sanger method of DNA sequencing, the chain termination, what we're really talking about here is DNA replication that is going to be interrupted at various points. In other words, it's going to be terminated. All right, the chain of nucleotides that are being replicated are going to be terminated or interrupted. So that's what we're going to do. Now, I'm going to come over here to this little panel and note that I've got two images representing nucleotides that could be used in DNA replication. Now, this first one here is called a deoxyribose nucleotide. Let me come over here to my pen, grab a color. So this is the sugar part right here, right? That's the deoxyribose sugar. And I've also got this thing here, that's going to represent a base, some kind of nitrogenous base, adenine, thymine, cytosine, guanine. This is the phosphate group right here. But the part that we're really interested in is down there. This region right here is the third carbon on the sugar. There's a carbon right here, and right here. And there are other atoms that are not drawn on this illustration, all right, just to keep it simple. This part of the sugar is the 3' carbon, and as you can see, there is a hydroxide group right there. Now, this is a deoxyribose nucleotide. Now, in DNA, the sugar is deoxyribose, right? Because it's deoxyribonucleic acid, as opposed to RNA, which is ribonucleic acid, in which case the sugar is ribose. But this is deoxyribose. And what we're getting at is this spot right here has lost an oxygen, or never had an oxygen. It was made that way, all right? So this is a deoxyribose sugar. But if you look down here, this next one has all of the same parts. We've got a nitrogenous base, we've got a phosphate, we got our sugar here. But you'll notice in the 3' carbon spot, there's an oxygen missing here. This is not a hydroxyl group, this is just an H. So not only was an oxygen not present on this group right here, making it deoxyribose, it's missing an oxygen here as well, making it dideoxyribose. These dideoxyribose nucleotides are very important for the Sanger method of DNA sequencing, all right? And I'll show you why in just a second. When DNA is being replicated, all right? So imagine, here, I'm going to come down here actually, and I'll show you what I'm talking about down here. All right. We've got a strand of DNA, and it's time to do DNA replication. Recall how the DNA double helix unzips like this, and then DNA polymerase starts adding on nucleotides along the chain, creating the complementary half, all right, using complementary base pairing. So if DNA polymerase saw this unknown DNA sequence or strand of DNA, and there was a C here, it would click a G on, right? And if there was an A here, remember we're making DNA, not RNA, it would click an A there, or if there's a T there, it would click an A on, and then a C, and then a T, and then a C, and then an A, etc. Basic complementary base pairing. Now the thing is, however, in fact, let me erase those and come back to this. If I put a G on here, all right, in order to add the next nucleotide in this spot right here, the adenine nucleotide, that would have to go there, there has to be a bond between this G and the A, okay, right here. They have to link together. Essentially, that is what I'm showing right here and right here and here and here. That bond links the nucleotides together to make the backbone of the DNA. Otherwise, all you've got is the hydrogen bonding going on there and the hydrogen bonding going on there. But the nucleotides need to attach to each other to actually make the strand of DNA. Now that bond occurs over here between the hydroxyl group of one nucleotide and the phosphate group of another. All right? The phosphate group from the nucleotide has to attach to the hydroxide group here, or the hydroxyl group. Now, I'm going to illustrate that for you over here. Okay, so here we have a typical deoxyribose nucleotide, and it's got the hydroxyl group here. And here we have another one, and I want to add this one to this one. I want to link them together. Now watch what's going to happen between the hydroxyl group of this one and the phosphate group of this one, okay? I'm going to bring this up, and as you can see right there, the hydroxyl group turns into an oxygen. This is the actual bond then between this nucleotide and this one. There's a condensation reaction, all right? You probably studied those. And the hydroxyl group has been changed. There's an oxygen, and these are linked up. Now, I want to add another nucleotide. I'm not talking, by the way, about the hydrogen bonding between, you know, the bases on this side. I'm talking about actually making the strand of DNA, the polymer of DNA. So if I bring up this next one here, again, the hydroxyl group is used to link the nucleotides together. And this is critical because, I'm now going to clear this up. Come back over to here. If we have a look at this dideoxyribose nucleotide, the dideoxy, note again that the hydroxyl group is gone right here. All right, there is no hydroxyl group here. It's just an H. In other words, any nucleotide coming up and trying to bond here will not be able to do so. And if this nucleotide is therefore incorporated into DNA replication, it will stop the rest of the replication process from occurring. Once it's added to the DNA chain, let's say it was added over here, once that happens, no other nucleotides can be added beyond that because there's no hydroxyl group there. So these dideoxy nucleotides, we can call them, are critical in causing chain termination. So there's the first concept you need to understand. All right, now, there are 4 different dideoxynucleotides. There is one for guanine, as you can see here with the G. So I've colored this in blue. I'm going to keep my color scheme going. Again, we can see the hydrogen right there instead of a hydroxyl, dideoxy. All right. There is a dideoxy analog. Okay, they're called analogs because they're similar to the other nucleotides. So like analogy, these are called analogs. There is a thymine analog, dideoxy analog, and a cytosine. Now, I should say that these nucleotides, when they're being used and added to the DNA chain, are really nucleoside triphosphates, all right? But I'm forgetting those other phosphates and just dealing with the typical nucleotides, and that's fine. All right, so we are going to be using these to create the replicated strand, keeping in mind we don't know this sequence. We want to figure out what are the bases in this sequence. We are going to use these dideoxynucleotides along with normal nucleotides and DNA polymerase, and we are going to perform DNA replication. All right, so I'm going to show you how that can be done. All right, so in terms of our experimental setup, and I'm keeping this very simple here, we are going to have 4 reaction vessels or 4 test tubes here. Each test tube has been color-coded to sort of match the dideoxynucleotide involved. And let me clear that up. You'll notice the sort of shapes I'm using for the nucleotides and the sugar phosphate backbone. I'm trying to use similar shapes that would click on and it would look like a ladder-like structure. So in this test tube, we are going to place the unknown DNA sequence from the virus. So here it is. It's in the test tube. We also have samples of that in this test tube and in this test tube, and you're not going to be able to see that color on there, but in that test tube as well. All right, so we have the unknown DNA in the test tubes. But in addition to that, we have DNA polymerase. Well, we need that enzyme to do DNA replication in all of these test tubes. We also have a bunch of normal nucleotides, and by the way, down here, those are illustrated in the same color as this. All right, just the cytosine ones, the thymine, the adenine, the guanine, the regular nucleotides that you've seen in DNA replication. But we also have those dideoxynucleotides, and those are the ones that I've got in color down here. Same color as here. So these are really dideoxyguanosine triphosphate, dideoxyadenosine triphosphate, dideoxythymine or thymidine triphosphate, I'm not sure, dideoxycytosine triphosphate. Essentially, these are the dideoxynucleotides. So you could just say it's guanine, it's adenine, thymine, cytosine, all right? Okay, now, in normal DNA replication, I'm going to reveal this DNA sequence again. Oops, yeah, a bit of a problem with that monitor. Probably doesn't affect the viewing, but it affects my use of it. In normal DNA sequencing, if there was a cytosine on here, a guanine would come up and hydrogen bonding would occur. This is complementary base pairing, and we would have our first base clicked in there. If there was a thymine here, an adenine would click in, and a guanine here, and a cytosine would click in. Now keep in mind that although I'm not drawing these, connected together, but they really would be connected together. I'm in a little bit of trouble here. Okay, they would be connected together, and that's critical in creating the DNA chain. Okay, we're having some trouble with the pen today. All right, let's see if I can grab another one of these, stick it in here. Okay, so this would create a DNA chain all the way along here with the backbones connected, and everything would be great. Now, here is the basis of the Sanger method of DNA sequencing or chain termination. Let me do it this way. Let me just go back to the beginning. It makes it a little bit easier. Okay. I'm going to reveal the DNA sequence and show you how this would work. Let's deal with test tube, the first test tube right here with the dideoxyguanine nucleotides in there. All right. And all of the other stuff and the unknown DNA. So here's the unknown DNA in this sort of bluish test tube. Okay, the first base is a C. Now, we don't know that, but we got to know that for this little simulation. So a regular G could come along and click on here, and that would be fine. And then DNA replication would continue, and A would come along, and these would bind up okay. A regular cytosine would come along, a regular thymine, and a regular cytosine, regular adenine. Now, you can see there's another C here. I could have a regular guanine attached there. Let's do that. A regular guanine could attach there. thymine, thymine, cytosine, adenine, and if a regular G was to attach to this point at the very end, I can't seem to move those very well. If a regular G came along at the very end, then we really wouldn't have accomplished anything special. We would have replicated DNA. There was no chain termination. But you know what? These nucleotides in the test tube, the normal ones, are not radioactive. We couldn't even see anything, and we really don't care about that, because if instead this one, the dideoxynucleotide attached, we now have a radioactive nucleotide in there that I can't seem to move very well. All right, a radioactive nucleotide, and now this means something. Now, what I'm saying is there is a long chain of replication. There was no termination that you could see because this one came in at the end. I'll show you something in just a minute. But what we have is 12 base pairs of DNA. That is a big piece relative to what I'm about to show you. So let me do it this way by going back to the beginning and to the next frame. It's a little easier instead of dragging everything around. Let me start again. And instead of a regular G going on there, what if this G, the dideoxyguanine, went on right there? Well, because this doesn't have the hydroxyl on the 3' carbon, no other bases. In other words, this adenine that would normally come in can't bond. And DNA replication is stopped right at that point. It's terminated by this dideoxy analog. Now, what this means is, and remember, we can't see this other stuff, but a guanine is in there. Now, we don't even really know a guanine is in there. We're going to have to do gel electrophoresis in a minute. But pretending we can see, no other nucleotides can add at this point. And we have a fragment of DNA that has the original sequence, fine, but only one base pair on it, a very small piece of DNA. Whereas, if I move that out of the way, imagine a regular G came on, and these are all happening in this test tube, all right? All the different combinations. An A came along, a C came along. Of course, they're being added by DNA polymerase. A T. Oh, we now have, where am I going? We now have another G, sorry. We'll add our C there. We add our A there. Ah, here's another C. So it's calling out for a guanine. But what if the radioactive one went on there, the dideoxynucleotide? The next nitrogenous base or nucleotide could not attach. The DNA replication is terminated at that point. But we have a different length fragment than when the G was on at the beginning. All right? We now have 1, 2, 3, 4, 5, 6, 7 base pairs and a little bit of extra, piece right there. But we have a bigger fragment. And if you know about gel electrophoresis, if perhaps you've done something on restriction enzymes or DNA fingerprinting, you know that gel electrophoresis can separate DNA based on the fragment size, all the way down to 1 base pair difference. I'll show you that in a minute. So in this test tube, we also have this variety. And that's about it for this. So let me start over. And let me show you what might happen in the second test tube. With all of the same stuff, okay, all the normal nucleotides, the DNA polymerase, all those things you need, plus the radioactive dideoxyadenine analog nucleotides. Okay, so we have our unknown strand, we start DNA replication, a G comes along, and now there's a T and an A would be added, correct? Now it could be this A, but if it was the dideoxyadenine right there, chain termination occurs, and you now have a fragment of DNA that has just 2 base pairs. But if a regular thymine was added there, we would go on, if a regular adenine was added there, we would go on, a cytosine would come along here, a thymine would be added here, another cytosine, and DNA replication is occurring, and now we hit another T, and now the dideoxynucleotide attaches there. So we've got a bigger fragment of DNA, and there will be all of these different fragment sizes in the test tube, chain termination occurs, and now this one is 6 base pairs. None of them will be the exact same length, okay? They're all going to be different lengths based on where the dideoxynucleotide attached. What if it attached way up there and all of these bases were filled in? This last one couldn't go on. Chain termination occurs. We now have, assuming these ones were in there, 11 base pairs. That's a different size DNA fragment. And this is the concept for all of these test tubes, all right, with this unknown DNA sequence. In fact, I'm going to show you now all of the possible fragments we could get if this was the DNA sequence and we're using these dideoxynucleotides. I'm just going to drag this over. Okay, so here we can see the different varieties. Let me move that down a bit. For the guanine dideoxynucleotide, the guanine could attach here on this first C, chain termination occurs, and now we have a 1 base pair strand of DNA with all unpaired bases for the rest. Or the guanine could attach on this cytosine right here, all right? And then these ones, there's chain termination right there. This is a 7 base pair sequence. It's a different fragment size. Or it could go right to the end. That's the biggest one we could possibly have, okay? Because it doesn't really matter what happens on that end, but that's it, 12 base pairs. If we look at the dideoxyadenine analog, it could attach on the second base right here. We have a 2 base pair fragment of DNA, or it comes down right to there where I showed you before, 6 base pairs, or all the way to the end, 11 base pairs. Notice these are all different sized fragments of DNA. For the dideoxythymine I didn't do, or the cytosine I didn't do for you, but you could probably imagine how it would work. All right, the T could go on here, all right? The T could go on there, or the T could go on there, and you get different fragment sizes every time. Same with the dideoxycytosine analog. C could go on the first G there, or on this G, or on that G, and every one of these is a different fragment length. Now, this is radioactive, so we're going to be able to see this stuff. We can also separate all these DNA fragments by gel electrophoresis, so we're going to have a look at that. Now, I think I can get rid of that guy. Just move that over there. Let's come over to here. Gel electrophoresis, we've got this gel substance set up. We've got an electric charge applied to the gel with the negative end here, the positive end here. Now, DNA is negatively charged, so it's going to be attracted to the positive terminal. It's going to get pulled through the gel. And the way that it works is the smallest pieces of DNA get pulled the furthest in a given amount of time, all right? Because it's light, it doesn't have as much mass, and it gets pulled through this gel, all right? It's not that it's moving downwards with gravity. This could be just laid on a table or whatever. It's just pulled by the electric charge. Now, I also have these lanes. I drew dotted lines, okay, on this gel. We've got these lanes set up for the test tube that had the dideoxyguanine analog. We're going to do something with this lane, and the dideoxyadenine, dideoxythymine, dideoxycytosine lane, they all have their own lane, like a little highway with 4 lanes. So what we're going to do now is allow DNA replication to occur and be terminated by the dideoxy analogs at various spots, but remember, in every test tube, there's only one type of dideoxy analog, so it's going to stop it at all of the G's, and this is going to stop it at all the A's. Now, I'm going to take this test tube, we pick up some of the sample, and we come over to the gel, we stick it in here, and empty the test tube or the pipette or whatever, and you're going to see these blue little lines, these rectangles. These represent fragments of DNA that travel different distances down the gel. The smallest fragments traveled the furthest distance. Now, let me just go ahead and take a sample of all of these and load them up into the gel. The red one goes into the gel. The green one goes into the gel. And the yellow one with the dideoxycytosine analog goes into the gel as well. And this is what we have. Oops. Okay. What you need to notice here is that every fragment of DNA, let me go to a different color, I'll just go to black. Every fragment is at a different position. This fragment went the furthest distance down. This one went the next distance down, next furthest down, and then this one went the next furthest down. Now, I need to bring over my fragment list over here so we can have a look at that as well. This is saying this is the smallest possible piece of the DNA in the test tube with the guanine dideoxynucleotide. So which one was that? Well, it's going to be this one, right? With one base. That means that the first base in the sequence of replicated DNA, this row, now not the original DNA, we'll come back to that in a second, but in the replicated ones, the first base was a guanine. And so we come over here and we write G. All right. In fact, I'm going to switch that over to a lighter color. Let's erase that. Whoops. I'll come over here. I'll write G. Okay. The next base in the gel that went down the next farthest was in the A lane, and so the next base is an A. Now let's see if we can find it over here. Oh yeah, well, we see that this one, there were two base pairs, so it makes sense that it went very far down the gel. And it's just one distance away from the one with one base pair. And so gel electrophoresis can separate just, single bases from each other. That's why it's so powerful. And we're basically going like a game, like a jumping, boom, okay, I've got to jump to the next level there, and then we're going to jump to that level, and then we're going to jump to that level. It's kind of a weird game, but that's the idea. So the next band of DNA is in the C lane, so that base is C. And the next one is in the T lane, so it's T, and then it's C, and then we just keep reading upwards. A, and then we're back over to the G, and then we have these two, so it's a T and a T, and this is getting out of line, a C, an A, and a G. Okay, sorry, that's a little messy, but this is what you do. You read upwards, all right? And, I mean, if we come to this last base here, G, go over to the G's, and you'll see that chain termination really went all the way to the end. I mean, the dideoxynucleotide went right over the end, and this is 12 base pairs long. It's the longest piece, and that's a G. And the second longest piece is 11 base pairs, and that's an A, and there's where the A is. Now, the thing to remember is this is the replicated DNA. It was interrupted every step of the way by the dideoxynucleotides. And that's how we were able to separate these into actual distinct fragments on the gel by blocking the DNA replication, by terminating the replication. Now, again, we are reading the order of these bases, G, A, C, T, G, A, C, T. We are reading the replicated DNA, not the actual unknown DNA sequence, which we see over here. Okay, let me clear that. This, right? That G, A, whatever, we're not actually seeing it here. So we have to take the complement. Okay? So in order to get the actual DNA sequence, just take the complement of this stuff. So if this is G, A, C, T, C, A, G, T, T, C, A, G, right? That's what we had before. We need to take the complement of that to get the original DNA sequence. Now, I'm going to do something here. I'm hiding this because we didn't know this. We put it in the test tubes. We let the dideoxy analogs attach and all the other ones attach, and then we put it into gel electrophoresis and we read it off there. And then we take the complement and we get this. We get C, T, G, A, G, T, C, A, G, T, C. This is the complementary DNA, and this is the original DNA that this was replicated from. So this is the replicated DNA that we read in the gel. This is the original. Does it look like the original DNA? CTG, AGT, CAA, GTC. Yes. All right. So I had to show you this at the beginning in order to show you how the technique would work. But really, you don't need it if you had this. If you had, for example, usually on a test or an exam, you're given the gel and you said, and you're told, deduce the actual DNA sequence and perhaps the amino acids coded for it. And we're going to do that in a second as well. And then you just read it off the gel. Just make sure that you're looking at which is the positive terminal. Now, you know, your prof or teacher, whatever, could trick you, could go positive there, negative there, and now you got to read from the bottom and it goes G, A, C, you know, T, T, you know, that kind of thing. So just be careful about that. But otherwise, you read the sequence in order and then you take the complement. Always remember, take the complement, because this whole technique of DNA sequencing is based on DNA replication that is interrupted or terminated by these dideoxynucleotides. So it's DNA replication. And what we're seeing in the Sanger gel here, all right, is we are seeing the complementary DNA formed by DNA replication. And you got to take the complement of that to get the original DNA sequence. Now, assuming you followed all that and everything is fine, let's figure out the amino acids. So I've got my little codon table here, all right? And I've got the original or the actual unknown DNA sequence. We have figured it out properly. I wrote it down here. I've erased it, but we've got it here. And you'll note that DNA is read in 3 bases. So each 3 bases in a row is a triplet in DNA, a triplet. But in order to figure out the genetic code and what the DNA codes for, DNA is transcribed and then translated on a ribosome into an amino acid sequence. All right. So we are going to have to convert, now that we know the DNA sequence of that original virus, we're going to convert this to mRNA, messenger RNA, and then we're going to use the codon table to figure out what amino acids were coded for by that unknown sequence. All right, so let's come down here and I'm going to write the messenger RNA. So G is the base that would be complementary to C if these were ribonucleotides. A, C. Now be careful, in messenger RNA there is no thymine, it's uracil. U, C, A. G, U, U, C, A, G. Okay, now, these guys here are codons. There's a codon. They're read 3 bases at a time. There are 4 codons here, and they would code for 4 amino acids. All right, so let's check out the codon table over here and figure out what amino acids are coded for by the mRNA, which was really coming from the original DNA. So GAC, so I come over here, here's the first base, G, and then over to A, so I'm in A, and C, there's my C. So there's the codon GAC. Aspartate is the first amino acid coded for by the DNA. UCA, so come up here, U, C, and then A, serine, amino acid 2 in the sequence. GUU, G, U, where's U and U? There we go. Valine, amino acid 3, and then C, A, G, glutamine, amino acid 4. So 4 amino acids were coded for by this unknown DNA. In order to figure out what the DNA sequence was, we had to use the Sanger method of, chain termination. We figured out the unknown DNA sequence looking on the gel. We then took the complement of that and we had the original DNA sequence. But we wanted to go a little further and say, hey, well, what could this possibly code for? That might be something that scientists might want to know as well. So we took this very short piece of DNA, all right, and we converted it to mRNA and then read off the codon table. All right, so that's basically how that technique is done. I hope you find that helpful. And I want to remind you that there are other resources that you might find helpful as well on my science website, www.zerobio.com. And of course, on my YouTube channel, there are other science videos as well. So hopefully you find that helpful and you understand what can be a complicated process. One other thing I want to mention to you, just to give you a bit more practice, I've got a worksheet for you that I will post underneath the video as well, that you can download right from my website here. And in this worksheet you've got some sample DNA replication that has been terminated at various points. For example, up here at the top here I've got a DDATP. All right, so I'm saying, well, at adenine, at that dideoxynucleotide, that's where termination occurs. So chain termination is there. And here, so we've got different fragment lengths again. This tube over here has DDCTP, so we're going to terminate at C or C. Now, you don't have these bases, do you? You don't know the strand of DNA, but using this sheet and then drawing, as it says down here in the instructions, draw the gel electrophoresis pattern, you will be able to deduce the original DNA sequence and then using a codon table figure out what amino acids are coded for. And one thing I will mention, by the way, is if there are any amino acids repeated, that indicates the redundancy of the genetic code. Sometimes there are codons that code, it could be 4 or 6 of them, that code for the same amino acid. This is called the redundancy of the genetic code. All right, so this worksheet will be available on my website, but I'll provide a link to it with this video as well. So hopefully that'll help you with the Sanger method. Okay, take care.